Welcome to the Weekly Neurology Deep Dive. This deep dive is produced by Amer Gavanini, and, we are just thrilled to have you joining us today.
Absolutely thrilled to be here.
So, before we jump into the data, I explicitly need to state that this audio is strictly for educational purposes.
I mean, there are absolutely no commercial goals here.
Exactly. It is only a summary. So, we highly recommend that you read the full paper to get all the critical details for your own clinical practice.
Because today, we are diving into a really fascinating paper.
We are. It is titled Development of a Diagnostic Autoantibody Assay to a Consensus Motif for the Risk Prediction of Epstein-Barr Virus-Related Multiple Sclerosis.
And that is by McCutcheon and colleagues, published in Neurology and Neuroimmunology and Neuroinflammation in 2026.
So, to kick things off, could you provide a quick practical summary for our neurologists who are listening on the go?
I certainly can. So, researchers have developed a highly specific consensus assay targeting a unique multiple sclerosis autoantibody motif.
And what were the main takeaways from that?
Well, this assay has a positivity rate of about 11% in multiple sclerosis patients. But most importantly, it can detect the disease years before clinical onset.
That is incredible.
It is. The findings strongly point to the Epstein-Barr virus protein BRRF2 as the initial trigger that breaks immune tolerance.
Which then leads to cross-reactive friendly fire against central nervous system proteins like vimentin.
Precisely.
So, thinking about how we currently predict multiple sclerosis, why could a new autoantibody assay like this change our daily clinical practice?
Well, you know the challenge. You have a patient sitting in your clinic with a radiologically isolated syndrome or a clinically isolated syndrome.
And you are looking at their MRI scans.
Exactly. You are weighing the immense risks of starting highly active disease-modifying therapies against the risk of doing nothing.
Because the eternal question is always who is going to definitively progress to multiple sclerosis and who will just remain stable.
Absolutely. Currently, we rely on MRI lesion load or oligoclonal bands.
Or maybe neurofilament light chain levels.
But predicting that definitive transition still remains incredibly difficult.
Yep.
So that clinical uncertainty is exactly what this paper targets.
By focusing on a specific signature motif, correct?
Yes. They looked at a precise sequence of amino acids, specifically a proline-serine-arginine motif.
Which is shared by hundreds of human and pathogen proteins.
Exactly. And the big data point here is that autoantibodies targeting this precise motif have been detected 5 to 6 years before a clinical diagnosis of multiple sclerosis.
It is almost like a very unique molecular lock.
I love that analogy.
For years, we knew multiple sclerosis patients were producing a key, the autoantibody, that fit this lock.
But we just needed to isolate that exact key to build a reliable diagnostic test.
So how did the researchers finally isolate it?
Well, they took memory B cells from a patient with multiple sclerosis. By activating those cells, they successfully isolated a monoclonal antibody specific to this motif.
Which allowed them to map the exact binding targets?
Precisely. They mapped the entire footprint and developed a robust, highly specific testing assay using consensus peptides.
So having isolated this monoclonal antibody and built the test, how well did this newly designed assay actually perform on real patient samples?
The data is really compelling. When tested on 179 multiple sclerosis patient serum samples, the new consensus assay showed a positivity rate of about 11%.
Which is actually an improvement from about 8% in an older multiplex version.
Exactly. The assay demonstrated excellent reliability, with a precision below 11%. But the most important metric is the specificity.
How specific are we talking?
Well, when tested on 91 healthy controls and 49 patients with completely unrelated neurologic diseases, the results were entirely negative.
Some absolute specificity.
Absolute. And they also applied this to longitudinal samples from the Department of Defense Serum Repository.
To look at retrospective prodromal testing.
Yes. The assay successfully identified multiple sclerosis signature autoantibodies in patients 2 to 8 years before their clinical diagnosis. And what was the signal difference?
Huge. The average signal strength for seropositive cases was around 577, compared to an average of just 24 for seronegative cases.
Wait, let me push back on this for a second.
Go ahead.
If the positivity rate is only about 11%, why should a practicing neurologist care about a test that technically misses about 89% of multiple sclerosis patients?
Your skepticism is totally justified, but, we have to conceptualize the heterogeneity of multiple sclerosis.
I see. So because it only captures about 11% of patients, the absolute specificity means A positive result is just a massive red flag.
Precisely. It is not meant to be a standalone diagnostic tool that completely replaces your clinical judgment.
Or your standard MRI criteria.
Exactly. It is more like a highly predictive puzzle piece.
Absolutely. You combine it with markers like neurofilament light chain or oligoclonal bands to create a definitive predictive risk score for early intervention.
And that early intervention is everything when we are trying to preserve axonal integrity over the course of a patient's lifetime. That makes total sense. But if this highly specific antibody is circulating in patients almost a decade before symptoms, something must be triggering it.
Which leads us directly into the viral connection.
Exactly. And we all know the heavily established link between multiple sclerosis and the Epstein-Barr virus.
I mean, the virus increases the risk of multiple sclerosis dramatically.
And almost all multiple sclerosis patients are infected with it.
So the researchers used the newly discovered monoclonal antibody to screen viral proteins.
And what did they find?
It bound with incredibly high affinity, about 1 nanomolar, to the Epstein-Barr virus tegument protein, known as BRRF2.
And it also bound to the viral glycoprotein M.
Exactly. And finding a high-affinity binding target within the Epstein-Barr virus proteome is a massive piece of evidence.
Because the Epstein-Barr virus produces so many proteins during an infection.
Precisely. So to compare this, they ran an enzyme-linked immunosorbent assay to compare BRRF2 against the widely known Epstein-Barr virus protein, EBNA1.
Because everyone usually focuses on EBNA1.
Exactly. The signal for EBNA1 was about 27% higher in multiple sclerosis patients compared to healthy controls.
But the signal for BRRF2 was about 43% higher.
Yes, and the differences for both were statistically significant.
So, while EBNA1 might cause a broader immune response, BRRF2 seems to be uniquely responsible for driving this specific multiple sclerosis autoantibody signature.
That is the crucial takeaway, and the paper added another incredible finding.
What was that?
Immunoglobulin G bands specifically directed against BRRF2 were detected only in the cerebrospinal fluid of patients who were positive for the multiple sclerosis signature.
Wait, so the antibodies were restricted entirely to the cerebrospinal fluid?
Absolutely, which places the immune reaction directly at the site of central nervous system damage.
But the Epstein-Barr virus is a virus, not brain tissue.
Exactly.
Why does an antibody designed to fight a viral protein end up causing demyelination in the central nervous system?
Well, this introduces the classic concept of molecular mimicry.
Where the immune system gets confused.
Exactly. The immune system is doing what it evolved to do. It generates autoantibodies to attack the Epstein-Barr virus during an acute infection.
But because the signature motif is also present in human proteins, the antibodies cause friendly fire.
Precisely. They cannot differentiate between the viral invader and the host tissue.
So to find out what the autoantibodies were attacking in the brain, researchers stained mouse neuronal cell cultures.
And what they discovered is that the dominant cross-reactive central nervous system target is a protein called vimentin.
Interesting.
Yes, the monoclonal antibody perfectly co-localized with vimentin and glial fibrillary acidic protein inside astrocytes.
It is exactly like identity theft.
How so?
Well, the immune system is given a wanted poster for the Epstein-Barr virus, but vimentin happens to have the exact same facial features.
I see where you're going.
So the immune system attacks the astrocytes by mistake.
That is a perfect way to visualize it, and there is a fascinating connection mentioned in the paper regarding that.
About infectious mononucleosis.
Yes. About 77% of patients with infectious mononucleosis develop immunoglobulin M antibodies against vimentin.
Compared to only about 9% of healthy controls.
Exactly. And since a history of infectious mononucleosis essentially doubles the risk of multiple sclerosis.
This vimentin cross-reactivity is the missing physiological link.
Absolutely. It connects the initial viral infection, the breakdown of immune tolerance, and the subsequent central nervous system inflammation a decade later.
The elegance of that mechanism is just staggering.
It really is. So, to repeat the practical summary for our neurologist listeners,
please do.
This paper details a highly specific consensus assay targeting a multiple sclerosis autoantibody motif.
Showing a positivity rate of about 11%.
Exactly. It reliably detects the disease years before symptoms appear.
And the evidence strongly identifies the Epstein-Barr virus protein BRRF2 as the initial trigger,
which breaks self-tolerance, leading to a cross-reactive attack on central nervous system vimentin.
For you, the practicing neurologist, this is so incredibly relevant. I mean, while this assay isn't on the clinic shelf tomorrow, It represents the very near future of neurology.
Identifying high-risk patients during the prodromal phase will be completely game-changing.
Absolutely. It will allow you to confidently initiate highly active disease-modifying therapies before irreversible axonal damage ever occurs.
Preventing the fire before it starts, rather than just treating the symptoms.
Exactly. So I want to leave you with a final thought to ponder as you go about your day in the clinic.
What is that?
Well, if we now know the exact viral protein, BRRF2, that breaks immune tolerance,
And we know exactly what it cross-reacts with in the brain.
could the future of multiple sclerosis treatment move entirely away from broad immunosuppression?
That is a fascinating question.
I mean, we could be looking toward a highly targeted tolerizing vaccine that simply neutralizes this one specific monoclonal antibody pathway.
Leaving the rest of the immune system perfectly intact to fight off other infections.
Exactly. Thank you for listening to the Weekly Neurology Deep Dive.